In this video, we will read Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of living organism under the system proposed by Whittaker in 1969, wherein he suggested the 5 kingdom classification with Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, Bryophytes, Pteridophytes, Gymnosperms and Angiosperms under Plantae. Let us also look at classification within Angiosperms to understand some of the concerns that influenced the classification systems. The earliest systems of classification used only gross superficial morphological characters such as habit, color, number and shape of the leaves, etc. They were based mainly on vegetative characters or on the androecium structure. This system was given by Linnaeus. Such systems were artificial since they separated the closely related species, since they were based on a few characteristics only. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy, that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. Let's discuss the first group under Plantae, that is algae. Algae are chlorophyll bearing, simple, thyloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils and wood. Some of them also occur in association with fungi, that is lichen. They form lichen along with fungi, which is a symbiotic associations, and they also occur in association with animals. For example, on sloth bear, epizoic kind of habitat. The form and size of algae is highly variable, ranging from colonial forms like volvox and filamentous forms like Eulothrix and Spirogyra, as you can see in figure 3.1. A1 figure shows volvox, which is a colonial form, and A2 shows Eulothrix, which is filamentous form. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, means motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in case of Eulothrix, or non-flagellated and or non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. For example, Volvox and Fucus shows oogamous type of reproduction. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine, brown and red algae produce large amounts of hydrocolloids which are water holding substances. For example, algin are produced by brown algae and carrageen is produced by red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Glacilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Let's discuss the first class, that is Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplasts may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplasts. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually take place by fragmentation. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Cara, as you can see in figure 3.1a. The next class is Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms as represented by Ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment, fucoxanthin, present in them. Food is stored as complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algae. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium, called as oogamous species. The gametes are pyriform, pear-shaped and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus, as can be seen in figure 3.1b. In B1 here we can see Laminaria, B2 Fucus, B3 Dictyota. All three, Laminaria, Fucus and Dictyota, are brown algae and they have similar structure, holdfast, stipe and frond here. In A1 we can see volvox and A2 we can see Eulothrix. Both A1 volvox, A2 Eulothrix are green algae. After Chlorophyceae and Phaeophyceae, let's discuss the third class, Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization as well. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, as can be seen in figure 3.1c, Gracilaria and Gelidium. In C1 here we see Porphyra and C2 we see Polysiphonia, both are red algae. Let's see the comparative account of three classes of algae, Chlorophyceae, Phaeophyceae and Rhodophyceae. Chlorophyceae are commonly called green algae. They have chlorophyll A and B as their main pigments, store starch as food. They have cellulosic cell wall and 2 to 8 flagella are present and flagella are equal and apical generally. Habitat is freshwater, brackish water and salt water. Let's see Phaeophyceae, commonly called brown algae, have chlorophyll A, C as and fucoxanthin as the major pigments. Stored food is mannitol and laminarin. Cell wall has cellulose and algin. Flagella are generally 2 in number, unequal and lateral. Habitat is freshwater but rarely freshwater, brackish water and salt water. Third class Rhodophyceae, commonly called red algae, have chlorophyll A, D and phycoerythrin as major pigments. Floridian starch as stored food, have cellulose, pectin and polysulfate esters in their cell walls. Flagella are absent, they are non-motile. Habitat is fresh water sometimes, brackish water, salt water mostly. Now let's read about the second major division of Plantae, which is Bryophyta, Bryophytes, Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Figure 3.2 shows the common bryophytes. A and B shows Marchantia. In A, Marchantia is female thallus gametophyte. B is showing Marchantia but male thallus gametophyte. Marchantia is dioecious, so male sex organ and female sex organs are present on separate thallus, separate gametophytes. Figure C here shows Funaria. Both gametophytic phase and sporophytic phase of the life cycle of Funaria can be seen here. Gametophytic portion has rhizoids, main axis which is basically equivalent to stem, and leaves called phyllates. And then sporophytic portion has foot which is embedded here in the thallus, seta which is elongated, and capsule which is basically curved and divided into three regions. Cannot be seen here, but we will discuss it later. Figure D here shows sphagnum. It is monoecious, so both archegonial branches bearing archegonia and entheridial branches bearing entheridia are present on the same thallus. This is the gametophyte of sphagnum. Gametophyte of Marchantia can be seen in A and B bearing the sex organs. Female sex organ in A, archegoniophore bears archegonia, and male sex organ bearing entheridiophore is seen here in the male thallus of Marchantia. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called entheridium. They produce biflagellate enterozoids. The female sex organ called archegonium is flask shaped and produces a single egg. The enterozoids are released into water where they come in contact with archegonium. An enterozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division called as meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packaging material for transshipment of living material because of their capacity to hold water. Mosses along with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Let's discuss liverworts first. The liverworts grow usually in moist, shady habitat such as banks of streams, marshy ground, damp soil, bark of trees and deep in the woods. The plant body of a liverwort is thalloid, for example Marchantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in 2 rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by formation of specialized structures called gemmae. Singular is gemma. Gemmae are green, multicellular, asexual buds which develop in small receptacles called gemmacups or gemmacups located on the thalli. The gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or in different thalli. The sporophyte is differentiated into foot, seta and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Next group of bryophytes are mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of 2 stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs Entheridia and Archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum and Sphagnum. Funaria and Sphagnum we just saw in figure 3.2. Next division under Plantae is Pteridophyta. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. Let's see figure 3.3 now. As we can see in figure A, we can see Selaginella, which is a pteridophyte, and plant body is divided into roots, stem and leaves. These organs possess well differentiated vascular tissues. The leaves in Pteridophyta are small, called as microphylls as seen in Selaginella, or large, called as macrophylls as seen in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones as seen in Selaginella and Equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic, thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids, the male gametes released from the antheridia, to the mouth of archegonium, which is the female sex organ. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large spores and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilotum belongs to Psilopsida, Lycopsida, Selaginella and Lycopodium belongs to Lycopsida, Sphenopsida, for example, Equisetum belongs to Sphenopsida, and Pteropsida, advanced ferns, Dryopteris, Pteris, Adiantum belongs to the class Pteropsida. Next division under Plantae are Gymnospermae, Gymnosperms. Gymnosperms, gymnos word means naked and sperma means seeds. Gymnosperms are plants in which ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, they are naked. Gymnosperms include medium sized trees or tall trees and shrubs, as you can see in figure 3.4. In this figure we can see Cycas, Pinus and Ginkgo, which are common gymnosperms. Cycas here is a medium sized tree, Pinus is a tall tree and Ginkgo is like a shrub. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree, as we see in case of Pinus. However, in Cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. Their nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and the female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, the zygote develops into an embryo and the ovule into seeds. These seeds are not covered. Next division under the plantae are Angiospermi, the Angiosperms. Unlike the Gymnosperms, where the ovules are naked, in the Angiosperms or flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In Angiosperms, the seeds are enclosed in fruits. So, these are also called flowering plants which bear fruits. The Angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest Wolffia to tall trees of eucalyptus which is over 100 meters in height. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. As we can see here, this figure here shows a dicotyledonous plant and this is the plant showing a monocotyledonous plants. Monocots are generally called grasses as well. Summary of the chapter. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae are chlorophyll bearing, simple, thelloid, autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of food stored, algae are classified into three classes, namely chlorophyceae, phaeophyceae and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different type of spores and sexually by formation of gametes which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. That is why we call bryophytes as amphibians of plant kingdom. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The male plant body of a bryophyte is gamete producing and is called a gametophyte. It bears the male sex organs called antheridia and female sex organs called archegonia. The male and female gametes produced fuse to form zygote which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. These organs possess well differentiated vascular tissues. The sporophytes bear sporangia which produce spores. The spores germinate to form gametophytes which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovule into seeds. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons.